Respiration is the process that almost all organisms on this planet use to meet their energy needs. Organisms gain energy from the food they eat, which is chemically mostly made up of organic molecules. Plants, during photosynthesis, convert light energy into potential energy. This potential energy is stored in the form of atomic arrangement within the organic molecules which are formed in the process. Animals and other organisms use these organic molecules as fuel and break them down into simpler molecules to extract energy by the process of cellular respiration. This energy is used to generate ATP, which powers most of the cellular work. So effectively, energy flows into our ecosystem as sunlight and leaves as heat. Although very different in mechanism, respiration is in principle very similar to the combustion of gasoline in an automobile engine. Explosion of a gasoline tank releases a lot of energy, and yet that energy can't be used to drive a car for long. Similarly, ignition of glucose releases explosive energy, which the cell can't utilize for doing useful work. To solve this problem, engines ignite a small quantity of the fuel-air mixture repetitively. In the cell, this effect is achieved by gradually extracting energy from the fuel molecule using a potential gradient. Two concepts are essential for understanding how nature makes this happen. 1. Redox reactions, and 2. Role of NAD+ in electron transport. In general, an electron which gets to be closer to a more electronegative element will have lower potential energy. Consider, for instance, the inorganic reaction of sodium with chlorine to form ionic sodium and chloride. Sodium loses an electron, hence gains positive charge, and is therefore oxidized in this reaction. Chlorine, on the other hand, gains an electron, thus gaining an overall negative charge, and is hence reduced. With organic molecules, often the charge transfer is partial, that is, there exists only a change in the degree of electron sharing in covalent bonds. The reaction of methane with oxygen clearly demonstrates electrons shifting away from the carbon as the reaction proceeds. Thus, methane is oxidized to carbon dioxide. In dioxygen, electrons are equally shared, but in the product, electrons are closer to the oxygen of water. Thus, oxygen is reduced to water. Similarly, when glucose reacts with oxygen to form carbon dioxide and water, glucose gets oxidized to carbon dioxide and oxygen reduced to water. From this it follows that in general, organic molecules that have an abundance of hydrogen are excellent fuels because their bonds are a source of hilltop electrons whose energy may be released as those electrons fall down an energy gradient. Since nature doesn't want fuel molecules, which here we will consider to be glucose, to react explosively, it breaks them down into a series of steps, each catalyzed by an enzyme. At key steps, electrons are stripped off of glucose, and as is often the case with oxidative reactions, along with the electron, a proton is released. Thus, effectively, a hydrogen atom is released. An enzyme called dehydrogenases removes a pair of hydrogen atom from glucose and transfers 2 electrons and a proton to a coenzyme called NAD+, turning it into NADH. The other proton is released into the surrounding solution. NAD+ is short for nicotinamide adenine dinucleotide. It is a derivative of the vitamin niacin. NAD+ accepts electrons and hence acts as a reducing agent during this step of respiration. Electrons lose very little of their potential energy when they are transferred from food to NAD+. Electrons from NADH eventually reach oxygen via an electron transport chain, which respiration uses to break the fall of electrons into several energy-releasing steps instead of using an explosive one. The fact that hydrogen comes from the food molecules is noteworthy. For each molecule of glucose, the cell generates up to about 38 molecules of ATP, making respiration similar to the process of caching large denomination for smaller, more practical ones. Cellular respiration can be divided into 3 parts for clarity. They are 1. Glycolysis, a process in which glucose is converted to pyruvate. Second, the citric acid cycle. Pyruvates are converted to carbon dioxide, which completes the conversion of glucose to CO2. And third, oxidative phosphorylation. This part accepts electrons from the first two, most often via NADH, and at the end of the chain, water is produced from the combustion of molecular oxygen and hydrogen ions. Glycolysis literally means splitting of sugar, and that is exactly what happens in this cycle, which takes place in the cytosol. Glycolysis requires the cell to expend energy initially. It all begins with the glucose molecule, which is phosphorylated by an enzyme called hexokinase, producing glucose 6-phosphate. Phosphorylation refers to the process of transfer of phosphate group. In this case, the phosphorus group comes from an ATP molecule. This step makes glucose more chemically reactive. The coupled arrows used in the diagram refer to the transfer of phosphate group or even a pair of electrons, as will be the case later, from one reactant to another. Next, glucose 6-phosphate catalytically rearranges to fructose 6-phosphate, the catalyst being phosphoglucoisomerase. Now fructose 6-phosphate is phosphorylated again to form fructose 1,6-bisphosphate. The enzyme here is phosphofructokinase, and again an ATP molecule donates its phosphorus. This step deserves extra attention as it's key to the regulation of glycolysis. Phosphofructokinase, the enzyme, is allosterically regulated by ATP and its products. With phosphate group on either side, the molecule is ready to be split into half. Aldolase, an enzyme, cleaves glucose into two different 3-carbon sugars, dihydroxy acetone phosphate and glyceraldehyde 3-phosphate. The resultants are isomers. The enzyme isomerase catalyzes the interconversion of the isomers, yet the reaction never reaches equilibrium since glyceraldehyde 3-phosphate is constantly consumed in the next step of glycolysis. Eventually, both molecules get to the next step of glycolysis. This completes the energy investment phase. The payoff phase starts with two sequential reactions catalyzed by triose phosphate dehydrogenase. First, sugar is oxidized by the transfer of electrons and H+ to NAD+, forming NADH. This reaction is exergonic, and energy from this drives the second reaction, that of addition of inorganic phosphate to the substrate, increasing its potential energy. Inorganic phosphate comes from the cytosol, where they are always present. The result is 1,3-bisphosphoglycerate. The 2 represents 2 molecules in number, which originated from the splitting of fructose 1,6-bisphosphate. The enzyme phosphoglycerokinase converts an ADP into ATP during the conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate. Also, now the product has turned into organic acid from sugar in the previous step. It is in this step that 2 ATPs are formed, which compensate for the 2 ATPs that were used during the investment phase. Next, phosphoglyceromutase relocates the phosphate group which was left to form 2-phosphoglycerate. Now enolase results in the formation of a double bond by extracting water molecule, yielding phosphoenol pyruvate. This causes the phosphate bond to become highly unstable, ideal for the next step. Pyruvate kinase, the last enzyme of glycolysis, adds phosphate group from PEP, that is phosphoenol pyruvate, to ADP to form ATP and pyruvate. Further, the NADH produced too can be used in oxidative phosphorylation of ATP if O2 is present. More energy can be extracted from the pyruvate molecule in the citric acid cycle. Overall, this step produces 2 more ATPs, which evaluates to 100% interest to the 2 ATPs which were initially invested.